So today we will talk about the NK cells, which are the basically part of second line of defense and which is part of innate immunity, right? The immunity which is present by birth, basically is present by birth, and it is basically non-specific, and we have looked into these things in a previous video. So today we will talk about the NK cells, which are known as natural killer cells, right? Basically these are non-engulfing cells, like they do not do any kind of phagocytosis, means no phagocytosis occur, as we have discussed in the macrophages and monocytes and dendritic cells. They do basically, their basic function is phagocytosis. So here no phagocytosis occur. One thing to remember, that every nucleated cell has MHC1. Now what is this? MHC1 means major histocompatibility complex, right? But it is absent in, like it is absent, or we will discuss about this thing in later on in our videos. Like, let me give you an overview. Like, it is absent in our RBCs, it is absent in our neurons, right? And it is absent in the sperm cells. It is absent in these three cells. But okay, now let's talk about the function of this NK cells, right? How these actually work. Like I have said, that they do not do any kind of phagocytosis, no engulfing. It what is their basically mechanism, how they kill the pathogen. Basically, for example, this is a cell, right? And this is a infected cell. Consider the one pathogen is there and it has entered. So obviously when a pathogen entered into a cell, basically the cell, what it do on every, like as I said, on every nucleated cell there is MHC1, right? So what the cell does, it sticks the part of a pathogen, or you can say the antigen on MHC1. Now what, as we have like discussed in a previous video, that there are certain you can say chemical barriers, or you can say there are interferons. Basically I'm talking about interferons. An infected cell basically release the interferon. Now like interferon is a protein which are released by the infected cell in order to alert the surrounding cells, in order to alert the neighbor cells, so they can become alert and they can basically protect, they can produce such mechanism in so that they can survive from that antigen, or basically survive from that virus or pathogen, right? So they release basically interferons. Now these interferons basically go to the NK cells, like they give the signals to the NK cells that something has entered into the infected cell. Now what will happen, this NK cell will go into the cell, it will go and check that is there any kind of antigen, or something is like, something is fishy, right? We can say this, something is fishy in our immune system. So what will happen, basically this NK cell will check the MHC1 if there is antigen. Like as you can see over here, the antigen is present. So it means, like basically now the NK cells are confirming this thing, like are these interferons not giving signal about something else, are they giving about this thing? Basically these NK cells are here now in the surrounding of the infected cell in order to confirm that, like basically what's the problem, is it confirmed that the antigen, because of antigen has entered into cell, that's where the interferons were released. Now what will happen, as this antigen like it will recognize the antigen, it will recognize the antigen. Now what will happen, these NK cells will release perforins. Now perforins, basically these are the Basically, perforins are the protein, right? These are the protein which basically make pores in the cell membrane of the infected cell. You can say these are the pores, right? Now, after this perforin, then there is a release granzyme. Basically, granzyme is a enzyme or is a chemical which is released by NK cell. Basically, it will blast. The function of granzyme is to blast the cell. Now, when the granzyme will be released in the infected cell, the cell will blast like a bomb. Basically, this is a function. This is how the NK cells do not do phagocytosis. It uses this mechanism. Basically, on NK cell, like in the infected cell, there are certain proteins. This is known as MICA and MICB. Basically, MICA stands for MHC, like major histocompatibility class chain related to protein A, right? And this is MICB, which means MHC, major histocompatibility complex class chain related to protein B. Basically, these both proteins, these are expressed. These proteins are expressed to give kill me. Like it is expressed to give me kill me signals to the immune system. Basically, when these both proteins are expressed on a infected cell, it means that they are giving the signals to immune system that please kill me. So what will happen? NKG2D. Now what is this? It stands for natural killer group 2 receptor D. Basically, this is the NK cells, and there are these receptors NKG2D. They basically recognize this MICA and MICB. As we have seen that here, this NK cells have come over here. Basically, at this time, these infected cells have expressed MICB and MICA, which are released to give kill me signals to immune system. When the NK cells, like NKG2D, like natural killer group 2 receptor D, recognize these both proteins on the surface of the infected cell, it releases perforins. First will release perforin in order to make pores in the infected cell. Then it will release granzyme, which will blast the cell. So how this mechanism works, this is how NK cell do not do phagocytosis. It works like this, right? So I hope this is clear to you. If you have any kind of queries, you can ask me in the comment section. I'll be very glad to answer you guys.